Good morning, class. How are you this morning? Can you hear me?
Yes.
Yes.
Thank you. Thank you. So it's Friday, November 20th. We have 3 lectures left in this class. And then one exam and one final. So we're almost there. And we have one chapter to discuss. So we're going to be talking about signal transduction. And let's get to that. Let's get to that so that we cover the material. Okay. So what are we going to learn? So we're going to talk about general features. General features of signal transduction. Specifically, how a signal is transmitted using ligands and receptors. We already know in principle how small molecules interact with proteins, how proteins interact with proteins. We know the forces behind those kind of interactions. So these will be just examples, but the nature of these interactions, we already know that. So we'll talk about some of the main kinds of receptors which are involved. So receptors, sort of the word itself kind of makes sense, right? Something that receives a signal. So we'll talk about G protein coupled receptors, a very common type of receptor. We'll talk about enzyme link receptors, right? So there'll be a small molecule that may inhibit or activate enzymatic function. We'll talk about gated ion channels, right? So ion channels that may allow certain ions to pass through, say, a cell membrane, but our small molecule will serve as a gate, right? Open and close ion channel, and various physiological processes that are accomplished using signal transduction. So generally cells live in communities, right? So we are a multicellular organism, right? So cells communicate with each other, even though they each have a cell membrane, have separate processes going on, but they will not survive on their own. They have to talk to each other. They have to exchange information. They have to exchange nutrients, and they have different functions. So, these signals that the cells receive from the environment, they are, for example, antigens, right? So these will be immune cells which recognize antigens, hormones. So many cells in the body produce hormones for other cells to grow, neurotransmitters, transduction of a nerve signal, nerve impulse, perception of light, very important, touch, right? So for the cells in the skin, for example, pheromones. This is more important, for example, for insects, right? But also for animals as well. So and so these signals, once they are perceived by the host cells, right, they will cause some kind of changes. And so, for example, they can cause the cell to undergo differentiation, right? So, for example, if a particular tissue has already outgrown, if there are so many cells already present in it, so the cells can start to undergo differentiation, where basically a new tissue will be formed from an old tissue. Antibody production, growth and size and strength, sexual versus asexual, cell division. So lots of different functions will undergo change. All right. So let's talk a little bit about receptors, just general overview. So in general, a receptor will be membrane bound, could be soluble protein or protein complex, which exerts some kind of physiological effect after the natural ligand binds to it. So these kind of receptors, so we'll talk about G protein coupled receptors. For example, epinephrine receptor. So we know that epinephrine receptor is present in many different tissues, many different cells in the body, right? And in general, this system, epinephrine, or also known as adrenaline, they also known as adrenaline receptor. Let's write this down. So you can hear about this receptor using two different terms. Adrenaline. So this is responsible for what's known as a fight or flight type response. So for example, when there is a ligand present, epinephrine, for example, it will bind to various cells in the body, for example, and one physiological response would be to divert blood from non-essential organs, such as stomach, for example, to skeletal muscles, right? So for example, if you have to fight or flight, it's not really wise to consume energy on digesting food, right? You already have to use the energy that you have. Instead, it's wise to use the energy by muscles for the fight or flight response. So, you know, for example, these receptors are also involved in drug discovery, right? You know, types of drugs known as beta blockers, for example, used for blood pressure, high blood pressure, right? So that receptor also regulates blood pressure in the body. Again, as a way to basically bind into this receptor, it can generate high blood pressure as part of the fight or flight response. All right, so enzyme linked receptors. So one of the receptors we're going to talk about is insulin receptor, right? So we know that insulin is a hormone which is produced by pancreas, whose function is to bind to various cells in the body and make them utilize glucose. So when the glucose concentration in the blood rises, it needs to be utilized, right? And so we will see that this receptor is actually has phosphorylating function, right? So in fact, it will autophosphorylate itself. That's very interesting how actually different parts of a receptor will phosphorylate other parts of the same molecule. So, so these are tyrosine residues, right? Tyrosine residues. So we'll talk about this in more detail. So you probably know what disease results. So if your pancreas are not functional, if they don't produce enough insulin, what disease comes from that?
Diabetes.
Diabetes. And so, you know, there are two kinds of diabetes, right? So what kind of diabetes will that be? There's type 1 and type 2.
Type 2.
So does everybody agree?
No, it's type 1, isn't it?
Well, type 1 is autoimmune destruction.
Right. So unfortunately, type 1 is childhood diabetes, in which the immune system basically destroys the cells in the pancreas, which are responsible for the production of insulin. And there's also type 2, Type 2 is more relevant to signal transduction, actually. So here, insulin is produced and actually binds to insulin receptor, but something happens there's a signal transduction defect. So the insulin signal is not transduced in a cell properly, not utilized properly. That's type 2 diabetes. So diabetes. All right, so ligand-gated ion channel. So for example, in nerve signal transduction, so you know acetylcholine receptor, so found in central nervous system, peripheral nervous system, CNS, PNS, also referred to as ionotropic receptor. Ionotropic receptor. And so what happens here, so acetylcholine, now there's muscarinic and there's nicotinic acetylcholine receptors. There are two kinds of those. So, and the idea behind this is that they interact with ligand-gated ion channels. So these will be non-selective ion channels. So for example, they allow the passage of sodium, potassium, calcium. And so in the presence of the ligand, these channels are open and allow for the passage of these ions down their concentration gradient. In the absence of the ligand, they close and the ions are no longer allowed to pass through. So these will be ligand-gated ion channels. Integrin receptors, so also very important ones, quite different from the other ones. They basically mediate cell extracellular matrix adhesion. Cell extracellular matrix adhesion. Extracellular matrix. So these are basically one of the main function is actually a simple function of holding the cell attached to the extracellular matrix, right? So the cell doesn't really detach and go do its own thing. But also integrin receptors, they participate in signal transduction, okay? So they regulate cell cycle, organization of cytoskeleton, and so on and so forth. And they regulate a lot of receptor tyrosine kinases embedded in cell membrane as well. So they have lots of functions. And the final type of receptor we're going to talk about are nuclear receptors. So we talked about steroids, various steroids derived from cholesterol, for example, right? So, so here the ligands are steroid, and thyroid hormones. Thyroid hormones. Okay. So these ones, the primary role of this is to regulate gene transcription, okay, or gene expression, I should say. So basically, they will directly bind to DNA, right? So these receptors will directly bind to DNA. Directly directly bind to DNA. And basically regulate the expression of adjacent genes. All right, so so these 5 kinds of receptors we're going to talk in more detail, right? But I think it will be a good, this slide is a good introduction into various receptors and their different functions and structures. Do you have any questions? So those are receptors. Now the typical ligands, so the ligands for the receptors are, could be as small as ions. So for example, ferric ion, right? So ferric ion, you know, it's iron 3+, right? As opposed to ferrous ion, which is iron 2+. So, so there are a lot of ligands for, there are a lot of receptors for that. In specifically, I kind of mentioned to you about ionophores in the previous chapter, right? Ionophores. Does anybody remember what ionophores are? So those are receptors for iron, right? What do they do? Anybody remembers ionophores? They produce by bacteria. No?
Is
Don't
that the vesicle transport with like the little, like it's like a little, I don't even know, some sort of like vesicle, and it, that ion attaches to it and then it transfers into the membrane?
Yeah, so well, these are not really vesicles, they're just proteins.
Yeah, proteins. Yes, that's what I mean, not vesicle.
Yeah. So there are proteins which bind iron 3+, and they basically bring it inside the bacterial cell. So bacterial cell basically produces those, releases these in the environment. Those come back and bring the iron back inside the bacteria, and then are released again, and just basically keep going back and forth, bringing iron. So organic molecules, so adrenaline or epinephrine, which is the same epinephrine receptor we talked about, could be peptides, so insulin. Now, insulin could be a small protein or sometimes referred to as a peptide. So here we have insulin receptor, and also vascular endothelial growth factor. So now what's known as a VEGF. VEGF receptor, very important receptor on the surface of vascular endothelial cells. All right, basically it's necessary. Does anybody knows what happens if the vascular endothelial growth factor binds to VEGF receptor?
Is it vasodilation?
No. Just the key word here is growth. So it's a growth factor. What do growth factors do?
Cause growth.
Right, cell division, growth. That's right. So this is not necessary for a process known as angiogenesis. Angiogenesis. Basically the formation and growth of new vascular endothelial cells. And this is a very important process, obviously, physiologically, but also has been a drug target for a lot of pharma companies, specifically because tumors are heavily involved in the construction of blood vessels. Why do you think tumors are so interested in angiogenesis?
So they can metastasize through the blood?
Yeah, maybe. I don't know. Yes, they can metastasize. I don't know that that's smart to think to build them for the future. Sort of, I don't know if they have their own brains to to analyze these situations. The primary, sorry,
And redirect blood flow directly to the tumor.
sorry, say it again.
To redirect nutrients and blood flow directly to the tumor.
Yeah, the problem with tumors is that they need oxygen, they need nutrients, right? They cannot grow without those. And so, for example, if tumor starts to grow, a tumor, let's say, grows near a blood vessel, it can receive the nutrients and oxygen from the blood vessel. But once the tumor reaches a certain size, then that blood vessel is not enough. Is not enough. The tumor starts to starve. The tumor starts to starve, and not only that, the tumor becomes hypoxic. Do you know the word hypoxia?
Without oxygen.
Yeah, so not enough oxygen. So the tumors becomes hypoxic. They starve for nutrients, and they start building their own blood vessels. Basically, they in a process known as angiogenesis, and so they basically cause overexpression of these vascular endothelial growth factors. And so there are a lot of drugs which target this receptor. And to target a receptor, you basically need another protein, because oftentimes it's very hard to block a receptor with a small molecule. It's a protein, right? And so there are a number of drugs on the market to treat cancer. And some of them are antibodies. One of the common ones is Avastin. Avastin. It's an antibody which is used for cancer treatment. Okay. And specifically used for colon cancer, lung cancer, and even glioblastoma, which to me is still a puzzle, how it can cross the blood-brain barriers. But that's why it's so ineffective, because it doesn't cross the blood-brain barrier easily. Glioblastoma. So Avastin antibody. Actually, Avastin is actually, so if you think about it, so if it causes the growth of blood vessels for the tumor, the opposite effect of that is also, for example, you administer this with drugs, right? So the drugs will not be delivered to the tumor very well. So it's a double-edged sword, right? On the one hand, you will make the tumor starve. On the other hand, you will reduce the delivery of cancer drugs to the tumor. So it's always a double-edged sword, and you have to be a very good doctor to prescribe the drug regimen in such a way that to get the most efficacious drug regimen for treatment of a particular tumor. All right. So any questions? All right. So remember this VEGF receptor, one of the key receptors now in cancer drug design. All right. So this is a summary of actually some special features of signal transducing systems. So one of them is specificity, right? So you can see here, receptor has specific geometry, right? Specific shape. And so it has to be complementary to its specific ligand. So it will not accept other types of ligands which can generate unwanted effects. Now, amplification. So usually you have one molecule arriving, interacting with the enzyme. That enzyme can actually perform function perform reaction on some of its substrates. Those substrates can be enzymes by themselves. They can perform enzymatic function on their substrates, and so on and so forth. And basically from one molecule of a ligand, you can generate lots of molecules which will continue on and carry on the signal inside the cell. Modularity. So here we have multivalent affinities with diverse signaling complexes. So for example, so modularity basically usually basically it's I don't know if I fully understand modularity. Modularity how signal is transduced. So the more of these molecules are involved in the response, for example, you phosphorylate this one, it binds to that one. Phosphorylate this one, it binds to that one. The more of these are involved in the process, right? The more kind of modules are brought in, the more specific the signal becomes. Because if something goes wrong, then it must be a different ligand. If there is some defect, then the signal stops. So so modularity is sort of it's a way to make the original signal more specific, right? So basically to bring in more players and create kind of specific modules which will help in transducing the signal more specifically. Another feature for receptor is desensitization or adaptation, right? So when there's too much signal, so there is too much signal, too much ligand arriving, and the receptor is overworking, there's too much response. So there is a mechanism to desensitize the receptor, for example, right? Specifically, one of the molecules downstream can come back, bind to the receptor, and make it less sensitive towards the signal that arrives from the outside. And the signal transduction will slow down. Then integration, right? So you know that in central nervous system, that's how our brains function, right? So signals arrive from different directions, and then they become this one particular neuron, for example, will process them all and add them up. There's so much of X coming from this, so much of X coming from that. So the net X will be such and such. And then let's compare the voltage potential. There's potential coming from this direction, potential coming from that direction. Let's add them up, derive the overall potential change, and that will result in a response, right? Once the certain critical value of X or potential changes, a response will be generated. And there's also what's known as a localized response. So here, you can transmit your signal far away, let's say to distant sites. Alternatively, you can employ what's known as a message remover in such a way that the response becomes local. So it's a way to localize the response just into the immediate environment of the receptor, right? So we're not going to look at examples of this. Let me see if I can find examples of this in the literature. That's my homework. All right, you guys have any questions? So these are main features of signal transducing systems. So for specificity, so here's just one example of one of these features. So so when a receptor binds to a particular substrate, let's say substrate 1, right, to form the receptor substrate complex, So remember KD, dissociation constant. Now, do you remember for tight affinity, the KD needs to be large or small? So you want small. Small, right. So the smaller this number for a particular substrate, for a particular ligand, right? The higher the affinity and the more specific it becomes. So you want, where do you want this number? So 10 to the negative 7 molar. What is this? Millimolar, micromolar, nanomolar? What kind of concentration is this? Can you convert this to micromolar?
Yes.
So what is it? How many micromoles is this?
To the -6.
Say it again?
So, micromolar is to the -6.
Yeah, so what's this? So how many micromoles is this?
it would,
100.
No.
would it be to the -13?
No. So if you can, so if 1 micromolar is 10 to the negative 6, so 10 to the negative 7 will be 0.1 micromolar, right? .1 micromolar, or somebody said 100, so it will be 100 nanomolar. So does everybody agree? Yeah. So so you want these affinities to be in nanomolar region. So most drugs actually bind to the receptors with nanomolar affinity. So that's what you aim for. If it's not nanomolar, then the receptor becomes non-specific. Instead, when you, in fact, when you publish a paper, you know, I've been on through this situation many times when I submit a research paper and our compounds that we publish do not have nanomolar activities, but have micromolar. I always hope that the reviewers and editors will still appreciate the compounds, even though they're micromolar, and they return the paper, they don't accept it, and they say, no, your compounds are micromolar. There's a high chance they'll be non-specific, they will interact with other targets in the body, and so take your manuscript back and improve the structure of your compounds, make them nanomolar, and then come back. So been on the receiving end of this specificity issue many times. And so the key word here is nanomolar. You want your compounds to be nanomolar. But you can also have picomolar. Picomolar are 10 to the negative 12, right? So there are such complexes which are picomolar. And so those are going to be highly specific for their receptor.
Dr. Kaneko,
Yes.
wouldn't it be 1 micromolar? Because we're just moving one place over from 10, not 0.1.
So, okay, so 1 micromolar will be 1 times
10 to the negative 6.
To the negative 6. That's 1 micromolar, right? And then if you turn this into 0.1, this will be 10 to the negative 7. Sorry. Yes. Does it make sense?
Okay, yeah.
Yeah. All right. So, so this is a general overview of these kind of receptors we just mentioned. So these are G protein coupled receptors. So in general, the way they work is, so you have this receptor, which is waiting for its ligand to arrive. Once the ligand arrives, so for example, epinephrine, right? Once the ligand arrives, it binds to the receptor. The ligand binding event then is accompanied by G protein binding. So the G protein comes in and binds to the receptor. And there is a conformational change in the receptor, which will allow for the G protein to detach from the receptor and bind to some kind of other molecule which have enzymatic function. And so this enzyme then will produce another molecule, X, commonly referred to as second messenger. Second messenger. So this will be G protein coupled receptor. Now this is a receptor tyrosine kinase. So we talked about insulin receptor, for example. So the way this usually will work is, so it's in many cases this will be homodimer, right? And it will have tyrosine residues on both sides, and there is enzymatic function on both sides. So this will be the enzyme, let's say, and this will have a free hydroxyl of tyrosine. And so this enzyme will phosphorylate this free hydroxyl and result in a phosphate on the tyrosine. So as you change tyrosine to phosphotyrosine, then different enzyme can come in and bind to that. Actually, when we start discussing this in more detail, I'll actually draw you some structures so that you know on the chemical level how it works. So phosphotyrosine then becomes a binding site for many other enzymes. And for insulin, for example, one of the immediate molecules that comes in and binds is insulin receptor substrate, IRS1. IRS1, and results in enzymatic cascade. And then eventually the signal goes all the way into the nucleus and results in the transcription. So we're going to actually get a protein out of that. These are gated ion channels. So the ligand will come in, open the channel, and will allow for the ions to come in or go out. Or the ligand by themselves, if this is a steroid. You know that steroids, they don't need really any kind of facilitated transport or active transport. They lipophilic, right? They can cross the membrane by themselves, go straight across the nuclear membrane as well. So they have to cross the plasma membrane, nuclear membrane, go towards the nucleus, bind to the some kind of transcription factor. So this will be a transcription factor. Transcription. So you know transcription factors are molecules, proteins, which can cause the expression of particular genes. And so here we have the messenger RNA and a protein. All right, so this is a representation of these kind of receptors using the simple cartoons. All right, so let's look at G protein coupled signaling in more detail. Why do we want to look at this in more detail? Is because let me give you some numbers. So genome encodes for 800 G protein coupled receptors, GPCRs. There is some literature saying that there are over 1,000. So scientists cannot agree on how many of them. And so these are for different growth factors, for hormones, for olfactory receptors, smell, for example. And GPCRs actually are overexpressed in 20% of cancers. Why do you think GPCRs are overexpressed in cancers? What benefit do cancers get by overexpressing GPCRs?
More growth factors, so that's why
Yeah.
they grow so fast.
Yep, perfect. Yep. If one of the function of GPCRs is to bind to growth factors, then cancers love that. They want these growth factors. So in fact, 1/3 of all the drugs on the market target GPCRs. That's a pretty sizable function of all the portion of all the drugs. 1/3 drugs target GPCRs, G protein coupled receptors. And so they used to treat things like hypertension, anxiety, glaucoma, migraine, headaches, and many of them are actually orphan receptors. Do you know this term known as orphan receptor or orphan protein, for example?
Do they just hold it till the proper ligand comes?
Well, these are basically molecules for which we don't know what the ligands are. So orphan receptors are those receptors that we have discovered. We have discovered them, but we don't know what their ligands are. Right? If we don't know what the ligands are, we don't really know what their function is, what they do. And so we would love to develop drugs for them, but we don't know what these drugs will be for. Right? And so pharma industry are actually very interested in these kind of receptors because that can lead to new intellectual property, right? So if you have a known GPCR, the chances are that there's already a drug for it, or there's some other company that is working on a drug for it. But if it's an orphan receptor, you don't really, there's no point working on a drug because you don't know what the drug is going to be for. And so there's a lot of, this is one way pharma industry actually invests into fundamental research to discover the function of orphan receptors, because it gives them intellectual property space. All right, so now G proteins are heterotrimeric, alpha, beta, gamma. And okay, so let's look, we already saw this picture. This is, for example, prototypical G protein. Yeah, let's look at this G protein and we'll finish this lecture. So prototypical G protein, RAS. So here there is a X-ray structure. It's a co-crystal, so there's a magnesium in there. And then there is a molecule of GTP, but this is not exactly GTP. I think if we look at this, it says specifically in this structure, it's non-hydrolyzable GTP analog, G-diphosphate and H-phosphate. So let's draw this. So it's a G diphosphate NH phosphate. So what do you think that is? So non-hydrolyzable GTP. So you know what GTP is? So G protein is a GTPase, right? What does this mean? Something that is GTPase, what does it do?
Breaks down GTP.
Right. So it breaks down the GTP to GDP, right? And by doing so, it can actually, basically it hydrolyzes it. By doing so, it can change, it can make things happen, right? Basically, GTP hydrolyzes to GDP, and then a signal can be transduced or shut off. So here, if you have normally your normal GTP has this structure, so guanine, right? And then you have oxygen, phosphorus, oxygen, phosphorus, oxygen, phosphorus. So it's a triphosphate, right? So normally, when the GTPase has this bound to it, it will hydrolyze. It will hydrolyze the terminal phosphate to GDP, right? Now, I don't have much room here, but I'll try to squeeze this in. G, and then you have oxygen, phosphorus, oxygen, phosphorus, oxygen, plus inorganic phosphate, plus PI, right? This makes sense? So GTP, which is this one, becomes GDP. So the problem is, if you try to obtain a crystal of RAS with the GTP bound to it, what will happen? You will not be able to get this crystal because GTP will undergo hydrolysis to GDP, and you will get the wrong crystal. And so, so what scientists have come up with, they have come up with a way to actually, instead of using GTP, to use non-hydrolyzable version of GTP. Non-hydrolyzable. And how can you make a non-hydrolyzable molecule? So if you have an ester, what can you do to the ester to make it non-hydrolyzable? What will you turn the ester into?
An ether?
Sorry, say it again.
To reduce it to like an ether?
Ether is one way. Yes, that's done oftentimes. That's correct. In fact, this is something that was the subject of my PhD, to introduce a CH2 group instead of one of the oxygens to make it an ether-like. But this one will be an amide-like. So instead of the ester, phosphodiester, we're going to have a phosphoramide. So here we have an ester. Here we have an ester. But the last one, instead of the oxygen, we're going to have a nitrogen. And now this bond here, obviously, will make it non-hydrolyzable, because amides are more what happened? Amides are more resistant to hydrolysis than esters. Does this make sense? And so what you will do, you will use this molecule to obtain a co-crystal of RAS protein together with GTP, and it will be non-hydrolyzable, stable, and you can get an X-ray structure. So there are a lot of these tricks people utilize by using basic fundamental OChem2, organic chemistry 2 chemistry, right? So a very neat approach. All right, I think we're out of time. Now, are there any questions?
I have a quick question. Is the orphan thing going to be on the test, what you mentioned on the previous slide?
Well, what do you mean it'll be on the test? You just need to remember what it is. Basically, I mean, it's just a receptor for which no ligand is known. That's all that. If it's on a test, that it'll be that.
Okay, thank you.
Yeah. Any other? All right. Well, you all have a good weekend, and I'll see you on Monday and then Thanksgiving break.
Thank you.
All right. Bye,
Thank
guys.
you.
Bye.